attend or what the hearing, so we should click OK there. And certainly appreciate you joining us. And I think you're set to go. If you want to share your PowerPoint, we'll be, let's see, I got to end my show, I think. Okay.
Okay, thank you so much, Dale. Yeah, my Wikipedia is way out of date. I don't trust anything on that. You guys, thank you so much for having me out here to, well, not out here, virtually here to talk to you.
Side by side.
Yeah, screen set up here. Give me a second. I've never found an elegant way to do this seamlessly. So
there it is.
We'll just do our best. There we go.
Please take a moment to mute your microphone so that we don't get feedback and such.
Yeah, please mute them.
Thanks. Yeah. All right. Well, and also, I just want to say, you guys, I know that there's, you know, some high profile other programs going on right now. So I appreciate time. I know we can maybe we'll find it a nice respite from the stress of the hearings, or maybe we can all go back and watch them later. I don't know. Okay, so yeah, thank you very much. I really appreciate the chance to talk to you. I am sorry I'm not there in person. I would love to be, but that is not to be, unfortunately. So in fact, today for those of you who are wondering where I am, I'm in my office in the University of Kansas campus in Lawrence, Kansas. Home of the NCAA winners, Jayhawks. We're very excited this year. But I'm actually on the traditional lands of the Kaw, who are also known as the Kanza and the Osage nations. So for those of you who are not familiar with the history of Kansas, it's an area with a long, long history of forced migrations and relocations. Many tribes have been forced into this area, out of this area, through this area. And of course, that has had disastrous consequences to them. And our lab group is a large group. We have multiple principal investigators, multiple PIs, and we make a conscious effort to try to not just be familiar with the history, but also to more proactively, not just end with land acknowledgements, but more proactively advocate and support the sovereignty of Indigenous tribes and communities, and to give back in the ways that we can, not just through our research, but through outreach efforts, or, you know, for example, when we give talks, we try to donate, you know, our honoraria, or at least parts of our honoraria, to whatever tribal organization is, you know, there that we could potentially benefit. So happy about that if you like. There are 4 federally recognized tribes in Kansas today, and they include the Prairie Band Potawatomi Nation, the Kickapoo tribe in Kansas, the Sac and Fox Nation of Missouri and Kansas and Nebraska, and the Iowa tribe in Kansas and Nebraska. And so we think that it's important to understand and acknowledge places throughout the Americas, but it's only a first step. And of course, we try to go beyond that first step. All right, so in my presentation, I want to first acknowledge the Indigenous and non-Indigenous scholars whose work has informed my thinking on these issues. And this is not all of them, but this is a sampling of the people who really influence the ways I have come to think about genetics and tribal sovereignty and working in this space. And a lot of the people whose names you see here are members of the SING organization, this is their logo, the Summer Internship for Indigenous Peoples in Genomics, which is an organization that it does a lot of things, but one of the primary missions is to advocate for ethical genomics research with Indigenous peoples, and to very specifically train Indigenous students and community members in genomics and genetics methods and ethics and in issues, and to build capacity within tribes for doing this research themselves, or at least for working with scientists like myself, outside scientists, to know like, okay, here are some things you should think about, and here's things you might want to advocate for in designing research projects. So I am not a founding member of SING, nor am I an administrator or core faculty, but I am a participant as a faculty member as often as I can be. And I'm a huge supporter of them. And so if you know of any students or community members or any Indigenous peoples who have an interest in genomics or should know about genomics, let me make my little plug here. Try to get this on their radar, so that I think it's just a really worthwhile organization, and it's growing and growing and growing. So just want to make a little plug for them because I think they're amazing, and I have learned so much from them. Okay, so let's get to the talk. I don't have to tell you this because you guys are all very steeped in these issues and you think about this a lot, but I wanted to start somewhere. So we're all fascinated, all of us here right now are fascinated by the origins and the history of our species, and that includes the last step on our global journey, which is the peopling of the continents that today we call the Americas. And as you know from all of the talks that you've had in your discussions, that there are still many questions about this process. And I think in some ways it feels like, here in 2022, we know less about this than we did 50 years ago, right? And that's a good thing. I think that's a very good thing in some ways. But these questions that we have right now are pretty basic. Who were these first peoples? Why did they travel? How did they survive the conditions of the Ice Age? How did they adapt to new lands, new environments? When did they get here? And by what routes, right? These are all pretty basic questions that we're still trying to solve. And I think that one of the reasons that we're revisiting these questions again and again is because we have a new kind of data that has been introduced within the last, decade or so. And it's not just genetics data, but it's complete genomes, the genomes from all of your chromosomes. And that has really changed the story. And that has revealed so many new details and continues to reveal so many new details every year that it's hard for me to keep up, let alone, your average non-scientist on the street who happens to be interested in this topic. And so that's why I wrote, part of the reason why I wrote the book that I did was I wanted to, first of all, really make sure I got all of this because it is my research, but I wanted to make sure I understood the history of it. But also, I wanted to give people a framework for understanding the field and understanding the issues and understanding the history. And I wanted to do it not just, my assumption is that my book will be out of date within the next 6 months, right? Some new discoveries are going to be just found or revealed, and we're going to have to revisit our models again, or at least add new details to them. But my hope is that my book can provide a framework that people who are not scientists and not specialists in this area can use to understand where we are today. And then, that will help them contextualize new information as it comes in. Okay, so let's get started into it. All right, so there are, as you know, many approaches to studying the past, and none of these approaches gives us a complete answer by itself. So there are, of course, many Native American societies that have histories of their own origins that have been transmitted mostly through ancient oral traditions for many generations. And some of these align with scientific ways of studying the past and some do not. And one of the things that I have concerns about is that a lot of my fellow scientists do not acknowledge traditional knowledge or see it as valuable. I want to say, I shouldn't say many, some scientists do not say that. I think it has become increasingly evident more and more that quite often you can find alignment between scientific perspectives on the past and traditional knowledge. That traditional knowledge can get us to places where scientific knowledge can, scientific approaches cannot. But even if they don't agree, it's still important, and it's still really important that people who work in this field are respectful and acknowledge that there are many ancient perspectives on these histories. Some of them are understood to be metaphorical. Some of them are literal explanations of origins, or are taken to be as literal explanations of origins, that peoples were always here, that they came from here. And I think that we who are not native should not dance around that or ignore it. We should be respectful and acknowledge it and position our own knowledge alongside it, not try to, create this hierarchy that has unfortunately been going on in the field a long time. I'm going to go ahead and admit people when they get into the waiting room because I'm getting notifications. Okay, so that is one of the things that I try to do in my book. And I want to say that, from a scientific perspective, events in the past unfolded in a particular way, but our understanding of the past is limited by the tools that we use and the data that are available. And as you know, I don't have to tell you this, archaeologists will look at the same sets of data and come up with totally different interpretations. And so it's not that different when you think about it, from having multivocal interpretations of the past. That is certainly the case. So, and I want to say that I take a perspective where I prioritize genetics data. That is my framework for working. And it often does not agree necessarily perfectly with archaeological data, nor does it necessarily agree with traditional knowledge. These are all just different ways of looking at the past and trying to come to an understanding. And that's where we are. So I'm going to kind of focus on genetics mostly in this talk, because I think you guys know a lot more archaeology, probably more than I do. So we're gonna kind of keep our focus on genetics. Okay, so I call myself an anthropological geneticist, and that's not necessarily a household term. So I usually try and define it when I give a talk. So anthropological genetics is a subfield of biological anthropology, which used to be called physical anthropology. And anthropological genetics is a synthetic field. So it applies the methods and the theory of population genetics to evolutionary and cultural questions of anthropology. And that can be questions in the present or questions in the past. And over the last decade or so, genomics, or the study of complete genomes, not just a single gene here or there, but complete genomes, these have become increasingly common in investigating human population origins, in population histories, and biological relationships among diverse human groups. And that is because the human genome is an archive of population history. So the evolutionary forces that act on all populations, which include natural selection, genetic drift, or random changes to the composition of gene pools of populations, mutation, which is the source of new genetic variation, and gene flow, which is, mating between populations. These change the genetic composition of populations. And this evolutionary history can be reconstructed using the tools of population genetics. Ancient DNA, or DNA from deceased organisms, is an extremely valuable tool because it gives us a direct window into the past. So we know that at this time, which we use radiocarbon methods to date, right? We know that these genetic variants were present in this person at this geographic location. But beyond that, we get that window, but beyond that, it opens up a window into the past, and all of that person's ancestors who've contributed to them genetically gives you then you can identify those contributions, and using population genetics you can reconstruct all of these things. So I have a direct message asking me to spotlight myself. I am not sure what that means, so I'm just going to keep on going unless somebody wants to to clarify that. Okay, trying to keep an eye on the chat as I go through here. So, all right. So in the case of the Americas, ancient DNA has helped us overturn old models, and it has yielded some really surprising insights into population histories. And we're going to get into that today. All right. I was going to skip this because I think you guys all know this, but let's just start at a baseline. So you guys all know the Clovis first story. This is what I learned in high school, or not in high school, well, in high school, but this is also what I learned in college. And in my book, I kind of talk about how I learned this story. And then literally the year or two later, I'm talking to my graduate, Frederica Kasel, and she's like, oh, nobody believes that anymore. And I was so confused. When I wrote my book, I was not going to start with Clovis first because I was like, nobody believes that anymore. Well, it turns out I'm wrong about that. There are plenty of archaeologists who still think that at least a late migration as Clovis first, not too far off from Clovis first, still think that holds true, right? A migration that, just predates Clovis, maybe 14,000 years ago, maybe a little bit earlier. There are some archaeologists who still buy into this, and I don't agree with that, but I did want to put it out there and do justice to their reasoning. And so the Clovis first model, if there's anybody here who doesn't know what I'm talking about, it's this idea that held sway in the field of archaeology for a very long time, 50 years, something like that, where that it was the Americas were peopled by a small group of big game hunter-gatherers who crossed the Bering Land Bridge very rapidly just before 13,000 years ago, and they moved down this ice-free corridor that opened up between the Cordilleran and Laurentide ice sheets, moved down there, raced down the corridor, and accounted for the sudden abrupt appearance of Clovis projectile points and Clovis toolkits that you see all across North America, almost all across North America, very rapidly around 13,000 years ago, give or take. And then a corollary to this model was that perhaps the American megafauna who went extinct at the end of the late Pleistocene were overhunted, right? This is the Clovis overkill hypothesis. So this was so entrenched. And again, it's what I learned in college. But of course, sites kept cropping up that didn't fit with this model. And so what do you do with those? For a long time, the archaeological community would dismiss them. But when a group of really eminent archaeologists went to Monteverde and they looked at the evidence and they finally mostly agreed that, yeah, this was probably a pre-Clovis site, that kind of opened the door to accepting a number of other sites, among them Paisley Cave, which also yielded genetic information, which was very interesting. And of course, that left these questions. So when did people first arrive? Well, we still don't really have a great answer to that. I'm going to give you a model and then I'm going to argue against my model. Okay, we'll get to that. Who were they? We kind of have a better idea on that one, genetically at least. So genetics has helped us help provide answers to a number of these questions. So let's talk about the genetics. I'm just going to focus on that for a while. And I guess really quick, I don't have a slide on this, but I kind of want to give you a quick rundown of the different kinds of DNA that we look at. So the earliest researchers in the field, among them my mentor, Dennis O'Rourke, who's now my chair at the University of Kansas, they focused mostly on, in the very, very early days, they focused on looking at what we call classical genetic markers. And so these are things like blood groups or, protein polymorphisms, things that you could get fairly easily with the available tools in the 1980s. Okay. And they would compare these classical markers between populations to kind of get an overall picture of how these populations were genetically related to each other, and did that match what people were reconstructing for morphologies or not. Then the tools of molecular biology began to be imported a little bit more precise, and we were able to start looking at mitochondrial DNA, which is DNA present in the mitochondria of your cells. mitochondria are little organelles in your cells, and there's hundreds, thousands of them in each cell, and they have their own separate genome, which reflects the evolution of the cell. It's a whole thing. We can get into that if you're curious. But their genomes are strictly maternally inherited. And that is because at conception, right, you've got the sperm coming in, you've got the egg. The sperm does not contribute anything but chromosomes, anything but that nuclear genome that we call it, the DNA that's in the nucleus, right? The egg is what provides everything else, all the organelles, which include mitochondria. And so that means that your mitochondrial genome, really small genome, it's only 16,000-ish base pairs, 16,400, whatever. It's small, but yours is exactly the same as your mother's and her mother's and her mother's and her mother's and so on, minus, with addition of mutations, a few mutations, over generations. It's a really useful tool to look at mitochondrial DNA because you can track the maternal histories of populations. And it gives you a good idea, using the tools of population genetics, you can create models and test models for population histories, how people are related, how populations are related. And it is very, very valuable, especially for ancient DNA, because it's so abundant relative to the nuclear genome, so abundant that you're much more likely to be able to recover it from ancient contexts. It's much easier to get than nuclear DNA. So when I started out as a grad student, that's what I was studying, mitochondrial DNA. But as I was in grad school, we were kind of going through another revolution, the genomic revolution in biology, and that quickly got ported over to the study of ancient DNA. And that was because they were now able to sequence the human genome, the whole genome, all the DNA from all your chromosomes. And that, unlike the nuclear DNA, gives you much, much higher resolution into the past. You can do much more powerful analyses. It's like, I compare it in my book to, with the mitochondrial DNA, you can get the edge pieces of a puzzle and you can kind of get, figure out what the picture is, probably, maybe. But with the nuclear genomes, you can get the whole puzzle. I mean, you're getting all those pieces. And so it's been extremely fascinating to watch the field develop, because in addition to better techniques for recovering ancient genomes, ancient nuclear genomes, they're also developing powerful and analytical methods for actually handling this massive amount of data to study this. Okay, so most of my talk is going to be focused on what we've learned from nuclear DNA and mostly from ancient nuclear DNA. Okay. I also want to stress, this is not my research. This has been done by many, many, many researchers in many different labs. All I did in my book was summarize all of this in, I hope, a way. So it's not stuff I'm doing. I am working in this field, but a lot of my research is not actually in this book because it's not published yet. So some of my research, but okay. So let's talk about what we have found as a field. All of the complete genomes recovered from the Americas so far have revealed that the first peoples of the Americas have two major ancestry components. One of them is from the ancient people of East Asia. The other major ancestry component was found through the sequencing of the remains of a little toddler who lived at the Malta site 25,000 years ago. Actually, I think he was there 24,000 years ago. In any case, this population was initially called the ancient North Eurasians. And then a little bit later, they sequenced genomes from two young people at the Yana rhinoceros horn site dating to 31,000 years ago, which is a really remarkable site. You guys may be familiar with it. This consists of there's a population living year-round above the Arctic Circle 31,000 years ago. It just blows my mind. It's incredible. And this population turns out is ancestral to this population. So in a sense, they're one population. You can call them the ancient North Siberians, as we call them in this figure, or you can call them the ancient North Eurasians, same, as far as I'm concerned, same people. So what Malta's genome shows us is that, let me slide here. Okay, here we go. So what Malta's genome shows us the ancient North Siberians and a group of East Eurasians had gene flow going on. And this interaction ended up forming a new group that became ancestral to multiple populations. And that includes present-day East Eurasians, and it also includes the ancestors of Beringian populations or the indigenous peoples of the Americas. Okay, so we're going to focus on this branch next. These present-day East Eurasians, by the way, are descended from the Paleo-Siberians. I'm not even going to get into that. Okay, so of course, all of this was taking place during the height of the last glacial maximum. And just to remind you, this was a period of global cooling and arid conditions reaching its peak between about 26,000 and 19,000 years ago. This is just a little cartoon that I found that shows you changes to the coastline over time. So here's the modern coastline. 21,000 years ago, you have this enormous land connection, Beringia. And this was a very harsh time in which to live. It was dry. There were, the conditions were very, very difficult. And around the world, the archaeological record and the genetic record too actually show us that populations are retreating into refugia in order to survive this period. And of course, the conditions of the LGM prevented people from moving very far into the Americas. Once that massive ice sheet was formed, it blocked passage out of Alaska until the opening of it along the coast, which, may have been available as early as 16,000 years ago. And then secondly, the next route would have been that interior route opening. A little controversial when that first opens, but, certainly before 13,000 years ago. Okay, so this is a figure from an article I did for Scientific American last year. So during the height of the LGM, we know that much of Siberia was depopulated, and we have evidence from the genomes of the first peoples that their ancestors were completely isolated. And so this is called the Beringian pause. It was first identified with mitochondrial DNA. One of the likely candidates locations for where this may have occurred was, of course, along the southern margin of Beringia, central Beringia, because paleoenvironmental reconstructions have shown that it was a much more hospitable place to live. And we, of course, don't have strong evidence, archaeological evidence supporting that. We do have a few, potential places that maybe show some, some hints that there might be people there during the LGM, but not a lot of archaeologists are on board with these yet. My colleagues here at KU are actually about to leave to go work in bluefish caves this summer. So we're hoping that maybe we'll learn more about it. We'll see. Okay. So during the LGM and during this period of isolation, several branches emerge from this ancestral population. One of them we know about from the genomes of the Upper Sun River children. I contributed to that project a little bit, and also from Trail Creek Cave. The individuals at these sites are actually part of a population from whom we have no present day descendants. We call them the ancient Beringians. And I don't think that's a great name, but that's what we call them. And they are closely related to indigenous, the other present day First Peoples. However, they don't seem to have direct descendants. And another branch that emerges during this time period, we don't know a whole lot about. We don't have genomes directly sampled from a contemporary member of that population, but they're called unsampled population A. We know about them from their contribution to the genomes of the present day Mixe in Central America. So that's an interesting unsolved question as to what is going on with unsampled population A. We do not know yet. And then the third branch that emerges during this period of isolation is, and this is between 22,000 and 18,000 years ago, give or take. The third branch is one which moves south of the ice sheets and becomes ancestral to all peoples south of the ice sheets. So we see the emergence of following that movement, and it's kind of showing here, we're going to now build on that map and move south of the ice sheets. Following that movement south, we see the emergence of two major genetic groupings from whom all Native Americans south of the ice sheets are descended. And this happens between about 17,500 and 14,600 years ago. The first grouping is called by geneticists Northern Native Americans, and that includes, it's abbreviated NNA, and it includes present-day Algonquians, Na Dene, Salishan, and Tsimshian speakers from Canada. And the second major grouping is called Southern Native Americans, or SNA. And that includes the 13,000-year-old Anzac child, who's the oldest known individual in the Americas, as well as Spirit Cave individual from right here, 10,700 years ago, and all present day and ancient Central American and Southern American groups from whom we have genomes. So they all belong to this branch. We don't know much about the United States, right? So a lot of times when I'm talking to non-scientists who are interested in this field, they're like, oh, Southern Native Americans, that just means people from South America and maybe Central America, right? I'm like, no, it includes, we think it includes North Americans as well. Well, we know it does because it includes these guys. But what about everybody over here? We don't know. Well, actually, I do know at least some, but I can't talk about it yet because we're not publishing our paper yet. But, you know, stay tuned for that. The reason we don't know is because a lot of tribes in the present day United States are not especially interested in working with geneticists on allowing DNA from their ancestors to be studied. And that is for very good reasons. We geneticists have not done a good job in respecting tribal sovereignty and respecting the sacredness of ancestral remains, and we haven't done a great job in building good trust relationships with tribes. And that is something that I and my colleagues, a number of colleagues at a number of institutions, are working very hard to improve. And it is working. We do have, you know, good collaborations going on, but this kind of research is very slow, at least the way I do it. And so that's why I can't talk about it yet. Sorry. But this huge gap in our understanding of genetics, genetic variation means, you know, that a lot of the models that I'm telling you today may be wrong. A lot of the dates that I'm telling you today do not account for all the genetic variation in the first peoples. And so we may see these things shift quite a bit as we add new genomes to the analyses. So this is what I'm telling you is based on the data we have today. But I want to acknowledge that we have some big gaps here. Okay, so in recent years, we, or I should say my colleagues, have been able to get genomes from dogs. And that has been really important because humans and dogs have an ancient relationship. We co-evolved as domestic partners during the Pleistocene. I mean, dogs were the first animals domesticated by humans. And, you know, I would argue they domesticated us as well. Of course, you know, this is why I put up cute pictures of cute dogs. You know, you get a cuteness response. That is exactly what dogs have done to us. So what's really interesting about American dogs is that genetic evidence has shown that they did not evolve independently from American wolves. They were domesticated in Siberia, most likely, and accompanied the first peoples as they moved into the Americas. And so therefore, it's a really important external source of data, right, to check our models with. And Angela Perry and her colleagues did a really wonderful study. And I believe that this figure was actually made, well, the original version of this figure, this is an adaptation by one of the illustrators from my publisher, but I believe the original of this figure that was published in their paper was done by David Meltzer, who's just brilliant. What it shows so nicely is how these patterns of branching between dogs and humans mirror each other so beautifully. And so what you can see here, these are all the dog genomes from Northeast Asia. These are the dog genomes from North America. Same with human genomes, right? And you can see that This is just mitochondrial DNA from dogs compared to whole genomes from humans, but I imagine sooner or later we'll get whole genomes from dogs as well. And I bet this pattern will hold up, but you can see this beautiful radiation of dog lineages that mirrors these Native Americans. Like here's ancient Beringians, here's NNA, Northern Southern Native American, Central Southern Native American, and Southern Native American. You see this splitting and rapid dispersal happening between 14.1 and 13.9 thousand years ago. And you see the same radiation happening beginning about 15,000 years ago by dogs. That is pretty convincing to me that, you know, our models for human populations are really good because the dogs are mirroring that. So that's great. I love this study. Love it. Okay. And so, oh yeah, so this split between SNA and NNA is happening between about 17,000 and 14,600 years ago. That's what this is. And the dog genomes kind of match that. Okay. Let's see, what else did I want to say about this figure? I think that's kind of it about this figure. Okay, so, and of course, archaeologists from Mike Waters' paper a while back, not all the pre-Columbus sites by a long shot, but archaeologists are identifying sites that predate Clovis, and the timing of these sites, you know, 14,200 years ago, maybe 15,000 years ago, is, you know, quite in line with the splitting of these genomes. So we have this really neat and tidy story. Here's Paisley Caves. Give a shout out to Paisley Caves. There we go. So then we have this really nice story, right? Everything seems to be working, lining up. So keep that in mind, put a pin in that. I'm going to bring us to a mystery in genetics, and then we'll do a little bit of a recap, and then I will argue against my own model here. Okay, so I talked about these different ancestry components. Now I want to talk about a very strange, mysterious one that we don't know really what's going on there, and that is this population Y. Well, first, unsampled population A, we have this drawn as sort of vague. Okay, we don't really know a whole lot of what happened after they, you know, split off from this branch and how they got to here. Maybe there was some kind of movement, who knows? Can't answer that one yet. So that's a mystery right now. And population Y is also a mystery. So we see population Y, rather Pontus Skoglund, geneticist who originally did this work and identified this signal, sees a signal of ancestry in South Americans, originally just from the Amazonian region, but since then has been found in other South Americans, including along the coast. And this is a signal of affinity with Australasian populations, which is just, right? Why do we see this relationship between Australasians, who I don't have a map on here, and South Americans? And everybody immediately goes to, oh, there was a trans-Pacific migration, right? No, it does not. The predictions for that do not match the patterns of population Y ancestry in the Americas, in South America. The model does not fit. In addition to that, let's see if I have this slide. Yes, I do. Okay. In addition to that, I'm going to argue against a trans-Pacific migration because, let me go back a few slides. Hang on. Sorry. Here we go. This individual, this 40,000-year-old individual from Tianwan Cave in China, has that ancestry too. So we would not expect that if it's just some Australasians migrating across the ocean, and then, you know, you would see a very different pattern. So there are two possibilities right now that we can't rule out. One of these possibilities is that this individual with this ancestry was part of a widespread population somewhere in Asia that became ancestral to the first peoples and also became ancestral to Australasians, and that this population Y ancestry got into the Beringian population and somehow, you know, filtered into the Americas. That is quite possible. Okay, so that's likely. That's kind of the one I but I go back and forth. The other possibility, I go back to my slide here. Whoops. The other possibility is that there was an earlier migration, that somebody was here, that there's a population here, and they migrated very, very early and got to South America very early. And then you have the main migration of first peoples that we see in the genetics data, and that as they came through, they encountered this population, had children with them, and we see the genetic legacy today. So we can't rule out either of those right now with current data. And I guess I should have taken a moment to explain this slide. This is a figure that's showing basically population relatedness. And so the darker these circles are, the more population Y ancestry they have, basically, the most closely related they are. Okay, so you can see that, you can see them in these. And then you just see kind of a scattering of it. And this is actually out of date because more individuals of population Y ancestry have been found here. Does anybody in the United States have this? We don't know. We're still working on that, trying to figure that out. We have no idea. But that's kind of important, right? That could help us get insights into this migration if we could figure this out. So stay tuned for that. All right, so let's recap because that was a lot. When did the peopling occur? This is a summary of the dates that we've estimated from genomes. And again, with the caveat that we're missing a lot of the population variation. So that these dates could change as we add in more genomes. Ancestral Native Americans diverge from Siberian and East Asian populations during the height of the LGM, at the beginning of the height of the LGM 25,000 years ago, plus or minus. Ancient Amerindians and other Native American lineages split from each other during this period of isolation between about 22 to 18,000 years ago. And then we see the division into two branches, NNA and SNA, of the peoples who are south of the ice sheets between about 17,500 and 14,600 years ago. There's actually a third branch that's represented by just one genome, but we're not going to worry about that right now. Okay, so I gave you a really nice, tidy story. I like that story. Let's talk about white sands. As you know, because you had a talk about this a few weeks ago, which I was unfortunately not able to come to, these footprints have been dated at white sands. They've been dated to the height of the LGM. They've been dated to between, 21 and 23,000 years ago, something like that. Not all archaeologists buy those dates. I was at the, I was actually a discussant at the session on the White Sands site at the SAAs this year, or this year. And there were a lot of questions about it, although it was not very contentious. I was kind of prepared. I was ready that there was going to be a big fight. There was not. It was all very, very cordial. But so some archaeologists dispute this. I cannot weigh in on this at all. I am not an archaeologist. I have no expertise here. So my question is, if we have this wonderful story that the genomes show us, and it seems to agree with the majority of archaeology, but then we get this thrown at us, showing us that people are below the ice sheets. They would have had to have gotten below the ice sheets 25,000 years ago, because that's when they start forming, right? How do we interpret this given the genetic evidence? I'm really thinking about that hard, because the one thing I don't want to do is just dismiss it, right? That's not in the spirit of science. So we need to incorporate this. And again, let me point out again that Indigenous peoples in this area are like, we have been here that long. This is not a surprise to us. It's really cool, but this is not a surprise. So how do we account for this? One possibility that I like, but I'm just waving my hands here. Maybe this is population Y, right? Or some other unsampled population that we detected genetically yet. And that is very possible because of that lack of genetic sampling that we have so much in the United States. So that is the thing I'm going with right now, but it's, we don't have any evidence to test this yet. It's a testable model. I'll just leave it at that. Okay. That was a lot of talking, and we're about to the end, and I wanted to leave time for questions. So these are ways to get hold of me if you're interested. I don't really update my website very often. In fact, it's been quite a while since I've written anything, but I mean to write more. My book, I go into all of this in a lot of detail. This is our lab website if you're interested in checking us out. We talk about the various projects that we've got. And these are my social medias if you want to, reach out to me there. If you're on Twitter, it's a lot easier to get a hold of me there than actually emailing me, because my email is a dumpster fire right now. I am very, very behind, as Dale can tell you. So yeah, thank you guys all for your attention. And I'm gonna stop sharing my screen and do some questions if you want. Dale, how do you want to do questions? Like, oh, you're muted. Still muted. I'm looking at the chat. I still can't hear you, Dale. Sorry.
Okay, David, I'm sorry. Anybody have a question for Jennifer? Certainly, this is the future, if you ask me, as far as how we're going to untangle all of the different problems. David, you must have a question. First, David, we got to unmute. You have to unmute. David, unmute. No, David, can you hear me? Unmute. I think you're mute. Hit the unmute button, David.
I have a question if we're waiting for David.
Yeah. Patrick.
Hi, Jennifer. Thank you. I'm a geologist, so I really appreciate you giving us the basics. And
Oh God, I hope I got it right. Don't tell me.
yes. No, I'm a geologist, so you got it right.
Okay, thanks. Appreciate it.
my question is a couple things. Well, one is where, for going forward for DNA work, where do you see the biggest the areas you'd like, the population areas you'd like to target to solve the biggest mysteries, the missing blanks,
Yes.
to give us a better picture.
Yeah.
And I appreciate your comments about building relations and so on. I look at recent geologic history of landslides, earthquake, and volcanism, and I've been working with I've had some students that were tribal members, and one of the things we found is interesting oral histories for geologic events that we're pretty sure go back more than a millennium, and the oral histories are still in their culture. So for me, I try to stay dialed into this rapport with them, because I don't want to publish things that they're not part of, being non-native. And anyway, so what you have thoughts on where you would target your research, or others collaborators would in the future?
Yeah, for sure. Yeah, and thanks for saying that. I could do a whole talk on collaboration and like how we go about it. And I don't not think that necessarily we're like the best or anything like that, but it's just it's important work to do. I think more conversations about how we can do this better, and the better, obviously. Okay, so my wish list for ancient DNA really, I've kind of already said it, but that present day United States, could we get some more ancient DNA from the Central Plains? I'm working on that, and the sort of Eastern woodlands. That would be amazing. I think my other pie in the sky wish list really would be, can we get DNA from the folks at White Sands? Now, of course, there are no burials that we know of. There's nothing to get DNA from, no like middens or trash heaps or hair or animal remains, nothing like that. So, and even if there were, would the tribes in the area be okay with genetics research? Possibly not, in which case, of course, we don't do that. So, but imagining that, my fondest wishes, if we could get DNA from them, that's what I would love to see. I would really love to know who was there. What was there? What did they look like genetically? I would also be very interested in getting DNA from folks in the Bering land bridge area, if we could do that, right? That would be amazing from the late Pleistocene. But we would have to have those those available to us, and that's not the case right now. So yeah.
Thank you.
Yeah.
Okay,
Are
other
you able-
questions?
Able to hear me now?
Yes.
Yeah, David, we can hear you.
I don't see a way of, oh, let's see. Can you hear me?
Yes. we can hear you
Okay.
fine.
One of the things I was really excited about is the connection you have with the White Sands, New Mexico, and all the footprints. I think that the ages of that site material that's recently come to light add a lot of information about your Beringians and when they came. And it's significant, I think, that they're as early as 21,000 years, which is before the Wisconsin glaciation. So you had people probably using water rafts or boats, canoes or something even at that time. That's exciting. When both Dale and I were students at the University of Washington, we took classes from Alex Krieger, who was a Paleo-Indian researcher from the mid-50s. He gave several presentations that's sort of a forerunner of what you were doing, in that he was looking at human populations, not just archaeology. And he considered the Amurians, for instance, as a group of people in the Kamchatka area and a little farther north from there, somewhere perhaps around 30, 40,000 years ago, as a very important dimension in the early peoples into the New World. The Clovis that you use is something that is kind of scarce and becomes even more so in the West, because it's mostly represented by kill sites of mammoth and other animals, sometimes sloth and others. But it turns out that the connections are much earlier in the intermontane West for the Western stemmed point tradition. That would now stratigraphically and chronologically be 3,000 or 4,000 years earlier than the earliest Clovis that we know of. Your example of Cooper's Ferry is a good one. That belongs to that tradition. Another would be Lind Cooley, which is also related to that. And so one thing that sticks in my mind too is Tom Dillahay and his work way down south in Chile. In that particular case, I don't know if you have spoken with him or not, but when he visited Dale and I in Seattle, one of the things he confessed is that he found an even earlier cultural occupation, but that since the whole site was in question by North American archaeologists, and they were having their review committee on the legitimacy of the site, that he wouldn't mention it, but he would write it up to document it. And of course, that turns out to be somewhere around 31,000 years. So that's something to reflect on in terms of just how early a lot of these people got into South America, and did they all come from the north? There are still good questions maybe about Africa and Asia that haven't been answered. Some of the ceramic wares that are so early in Peru, and they spread northward. So a lot of technology and food subsistence. I think we've recently discovered that maize coming into Belize, for instance, the origin is actually in South America. And so that came as a surprise, thinking that it might be more in Mexico or thereabouts. But another is that the fossil animals are another clue, because the late Pleistocene animals that we have represented indicate a northward movement from South America. For instance, at Tequixquiac in the reservoir north of Mexico City, there's a llama sacrum that was carved that's somewhere around 25,000 years old. So you see, there are some things that may be returning, and I'm implying here that it wouldn't apply only to animals. There may have been a northward resurge of earlier people in South America back northward once they reached Tierra del Fuego. So these I mentioned to you just because they give you a few ideas to think about and possibly incorporate. But I think that the basic model that you have is almost a blueprint of what Alex Krieger's thoughts were for the New World. He wasn't just a North American archaeologist. He spent a lot of time in Mexico and South America too. So thank you for a very inviting and thought-worthy exercise. I particularly like the title that you give yourself as an anthropological humanist. We need more of them, and I regard myself in some respects to being one of those too.
Thank you. Thank you so much for your thoughts. Yeah, I have no problem with populations moving northward. I think, especially through the ice-free corridor. I mean, that's of course much more recent, but you know, of course, the only evidence we have archaeologically of people moving in the corridor region is northward. So I disagree with a number of some of my Alaskan colleagues on that one. Okay, yes. So just to respond a bit about genetics. So I think that, yeah, we definitely see these earlier sites, you know, and I have corresponded with Professor Dillehay, although I've never actually gotten a chance to meet because we don't go to the same conferences usually. And, you know, I have no problem with these early sites. I know that a lot of my archaeological colleagues, though, find them to be less than persuasive. So, you know, I try and keep an open mind about them. The genetics, and again, I'll keep coming back to this as my frame of reference is the DNA, right? And the population, the biological population histories, and what those show us right now with existing evidence is, you know, this, you know, slightly later migration, more like 16, 17,000 years ago, but maybe population Y is the, and some other of these unsampled populations, the key to understanding that. I will say this, you know, you mentioned origins in different places. We don't see any biological evidence for that. So no biological evidence for an African migration whatsoever, no biological evidence for anything else. So if it happened, you know, we would have to see, I would have to see more, I would have to see DNA to convince me. I will then argue against myself, right? Because we know the Vikings were here, they left no genetic contributions. So, you know, it is possible for people to have been here without leaving a genetic contribution. But I would imagine, like with the Solutrean hypothesis, that if we're really positing a pretty big migration, significant technological transfer, we got to see some genetic evidence of people here. So my feelings are sort of bounded by the DNA at this point about that. But the other thing I wanted to say in response is Dan Odess is now, or at least in when I've been talking to him about this, and we've talked a lot about this, and maybe he mentioned, I don't know if you guys have had him speak to you about, but he thinks the White Sands, you know, this is such an early migration. He's now back to the overland route for those guys. So he's more convinced by an overland route 25,000 plus years ago than a coastal route at the same time. I'm not sure I agree, but you know, we'll just leave it there because I don't think we really have genetic evidence to speak to that yet. So yeah, that's all my thoughts, somebody said.
Other questions? I don't seem to see raised hands, but yeah, anybody?
Jennifer, I do have another question. Do you talking about materials and knowing that some of them are sensitive, obviously, well, they're all sensitive because they're tribal remains and so on. But can you talk about the quality of the DNA you get, might say, from hair, bone, and coprolites, or some? I mean, what are there limitations from one to the other? You probably talk about this in your book, which I've not gotten yet, but I do intend to buy. Looking forward to it, in fact.
Thank you.
Thank you.
Yes, there are limitations. And I guess, I mean, I think I didn't stress this enough, how few genomes we have, right? How poorly DNA is generally preserved in the past, in most contexts. It's really bad. So, I mean, I will say, in some, Arctic is a whole different region. I love working in the Arctic because the DNA is really, really good up there. But the rest of the America is not so great. And so you're really looking for the diamonds in the rough. And so what we have found, and again, I'm using we very loosely, I mean, my colleagues really have done most of this work, is that the best DNA these days appears to be preserved in teeth and in the petrous portion of the human cranium. Often for cultural reasons, the petrous portion is not a great thing to sample because you're drilling into the skull, and teeth can often be more appropriate to sample for some communities. And some will be like, no, we don't want you to do anything like that. You can just collect loose fragments of bone, or so it's whatever you've got available, then you work with, and it may or may not have DNA. And that's why our grants tend to be, I mean, it's expensive to do this research. It's really expensive. And so the approach that our lab takes, and I guess this is what other labs do too, I'm not sure. We're not a big lab. We don't have the resources that, Harvard or Copenhagen or, the big labs have. So what we do is we will, when we have permission to do research, we'll try to clean all of the ancestors to see who's got the most DNA, and then we focus on those. Unless we need to address some particular question with a particular ancestor, and then we'll try our best with that individual. But, yeah.
Patrick, I would also add, Patrick, that boy, you'll be blown away when you get the book about what they go through to try to get the DNA, and just the amazing amount of work. So it's well worth just getting at that idea, what the research have to do. One quick question. I'm a big fan of what Tom Higham did with the World Before Us book, and he comes up with DNA in dirt. I don't know, in the caves, in the dirt. Not like Patrick saying coprolized teeth or whatever, but in the dirt. What do they call it? DNA dirt or something like that?
DNA. Environmental DNA, or some call it SED DNA, sedimentary ancient DNA.
I think it's unlikely you'd see it at White Sands, right?
I don't think so, just given the conditions there. But maybe, I don't know. You never know with ancient DNA. You never know. But I'm a little pessimistic about that one, but we'll see. Maybe somebody will be able to do it. There are people in our lab group, not my students, but Dennis O'Rourke's students, who do SEDA DNA research, and it's really cool. Yeah, they're working on some really cool stuff, including, I think it's okay to say, a bluefish. I think they've been talking about those in public talks, so it's okay to mention that.
Yeah.
So for those of us who are not specialists, like what is the sort of threshold like?
Okay, so for us, and this is cost, right? So if you think about it, you can get, DNA. So you do an extraction, you do a DNA extraction in the lab, and then that extracts all of the DNA in a sample. So that includes the human DNA if it's there, but it also includes plant DNA, soil bacteria, every microbe and every human, a modern human who's touched it, right? That it's all co-extracted. And so then you have to fish out these tiny little fragments, these really scarce damaged fragments that belong to that individual, what we call the endogenous DNA, right? And so you can go back and keep sequencing over and over again and try to get every single fragment sequence, but it just becomes exponentially expensive to do, especially if it's not very high in quantity. And so we, as a not super wealthy lab group, what we do, we have a threshold. And so we'll do light sequencing, which will get us, you know, some thousands numbers of, look at how many reads, and those reads are literally individual fragments of DNA. They're very short. And we'll look at the number of reads, and we can tell based on damage patterns, because ancient DNA has very specific damage patterns. We can sort them out, human from, you know, ancient from modern, and then, you know, align them to the human genome and say, okay, we've got this number of ancient human DNA reads, and that is this percentage of the overall reads that we got. Okay, so you do that bioinformatically. And what we're looking for, if we want to get a whole genome, we're looking for 10% of the DNA extracted being endogenous or belonging to that ancient individual or more. That's if we want a shotgun sequence or just sequence all the genomes. If we have less than that, shotgun sequencing is not cost effective. It will be way too expensive for us to try to do, because you just have to keep sequencing again and again and again. And every time you do a sequencing run, it's really expensive. So luckily, okay, so very, very few samples have more than 10% endogenous. I think in one of the projects I've got like one, and even that's kind of iffy. Arctic, you get more. In the Arctic, you get more samples. But we're really, so that normally would kill any hopes of doing, you know, genomic projects for us smaller labs. But the Reich lab and his collaborators at Harvard and his enormous collaborative group, they have developed another method, which I won't get into the details because it's already late, but they've developed, essentially you can fish for these ancient fragments, you enrich for them, you pull them out, and with a predetermined set of probes that will target specific regions across the genome. And you can do that for samples that have, for us, between 1% and 10% endogenous. And so that opens up a whole world more samples that we can get at. So I hope that answered your question.
Yeah, thanks. Super great. Amazing cutting edge research, really.
I mean, there's some brilliant people working in this field. We just
this discussion that you have had here brings me back to another example for biological anthropology. That is back in the 1990s. There was an anthropologist, archaeologist in the Pacific Northwest and in Maine. Robson Bonnickson was his name. And he was involved with the Center for the Study of the First Americans. And he was working a site over in Montana, and at a clay layer, about 8 to 10 feet below the surface, they ran into hard clay and mammoth remains, and the mammoth produced a great deal of hair. So he became quite interested in the hair and wanted to know just how frail it was. And so he, in the process, discovered in his analysis that there were a few human hairs too. And so he did a large analysis to determine if the field crew was responsible or if it actually was archaeological. They concluded it was archaeological, and it was of a mongoloid family of some sort or another, based on what the typology was at that time in the 1990s. We had to stop the research at that point because he did find that it contained DNA, and he wanted to be able to take it farther and see, well, how well preserved was the DNA? Is it really comparative? He didn't have enough to work with. So I funded him to go to another place called the Yakima Training Center. It's an Army installation in Eastern Washington, out in the desert of the Columbia Plateau. There, he took samples of soil that archaeologists had excavated in level bags, and took areas that specifically used a technique he had for floating hair out of the midden. The value is that it was an open site, and we were able to prove that hair preserves quite well once it's calcified. For instance, it doesn't deteriorate. The faunal remains, and this is the interesting part from the archaeology, indicated about a dozen different faunal remains of mammalian animals. But the hair was compared with the comparative collections at Oregon State University for male and female mammals of Oregon, for instance. They were able to identify 39 species of mammals from the same sites. Now that's a real eye-opener. That would indicate that hair not only is preserved, it can be recovered, and it may be more informational about the range of mammals who are actually there than we get from the faunal analysis using osteology. That's worth considering. I tried it once further in Montana and near the Libby Dam. He also found all kinds of hair there for mammals, but no human hair. But again, he showed that there was a greater variety of evidence for the faunal setting that people lived in and brought to the campsite.
That's really
Thanks,
fascinating.
David. Yeah. Patrick, I just want to point out that you can get the book on Kindle or audio too, if it's a good audio. I've heard samples. Any other questions or response? We probably should let Jennifer, it's gotta be
Just past my bedtime.
10:30 and she has a 3.5-year-old.
I hope he's asleep.
Time
by now.
for one more question. One more, anybody? I certainly
I'll
appreciate
type
it.
my
And
I'll type my email into the chat, but I gotta tell you guys, I probably will not answer any emails for like months because I'm behind so badly.
It's the end of the term and she's been
Yeah. All right. There it is. Though. You have it. Don't be offended if you don't answer. I'm sorry.
I really appreciate, and I know we have tribal members in the audience, but I really appreciate the effort to try to develop trust, because it's so critical on tribes. If you really, really want to know these things too, usually, and you see that. But your book does a brilliant job of explaining how bad we've done, and it's going to take a bit to straighten that out in terms of this kind of work. No last questions? Well, I do want to thank you very much, and thanks for contributing to the Northwest Native American Basque Weavers Association. And we do appreciate you coming to the Northwest with this best-selling book, and I look forward to the next ones. I really do. And
I guess this is a good excuse for me to go to the SAAs, or good inducement to go to the SAAs next year, which I don't usually go, but I might go this year just to.
Yeah, I think we do have a last question. Do you want to go ahead? Maureen, or did you have a question? Looked like you had a hand up, but you just came in.
I was just clapping my hands.
Oh, good for you.
Thank you. So much.
Sorry, we came in late. Is this going to be a recording so we can watch it?
Yes.
Oh, good. Oh,
Yeah,
good.
she agreed to that, and I'm so excited for that, even though we don't have Matt, our president, here but he told me how to record it, so we should be just fine, Mo. And
Oh,
you've been watching the hearings, I bet. You got your apron on, maybe.
I know. I cooked dinner and now I'm making the bread. So
That sounds nice.
sorry, Jennifer.
Oh no, that's
Hey,
all good.
well, that
It's all
was
good.
our last question, but
All
thank
right.
you so much, Jennifer,
Well,
and have
thank
a
you
great-
all so much. I
Glad you're
hope I'll see you. I hope I'll see you.
doing
in
well
Portland.
at the end of your
Yeah,
term.
take care.
Thank
Thanks
you
for
all.
having
Thank you, Dale.
Look forward to
Bye.
seeing you in Portland.
All right.
Bye.
Bye-bye.
Thanks, Dale.
Yeah, thank you, David. That hair is a real interesting part of this.
We'll be in touch soon.
I hope we get more of that DNA work. Rob was amazing in that area. You're doing well?
Yes.
Good. Yeah, I mentioned, I don't know if you heard me, but we've been in touch with Kelso. They'll probably tell you. But they're going to the Museum Association meeting in I don't know where it is. It didn't seem to show, but they're going to talk to Charlene Christ, the director of the Squaxin Museum, about doing a little display of the bones we dated.
Oh, yeah.
And you're on the preservation committee, so we need your help now. So they will meet Charlene and Joe,
Okay,
and then we hope it to be you know, with teeth and bones, and we're getting Squaxin artists excited about doing some display work, and then travel it around to other tribal museums, you know, like the Suquamish and Tulalip Macaw.
good idea. Keep it up.
You may not have heard, I just talked to everybody and we're just getting them together for this meeting at the middle of the month. Thank you. Have a good evening, and I'm glad we had an audience with those hearings starting today. But we'll see a lot of that, don't worry, you'll see it. Bye-bye now.
Bye now.
Glad you're doing well.